Module 5, Objective 9, discuss the two types of postsynaptic potentials, EPSPs and IPSPs, and how the process of summation generates or inhibits an action potential. All right, information processing. So we've started this module talking about the resting membrane potential and a graded membrane potential, and we know that those all kind of are found within the cell body. We know that the action potential is propagated down the length of the axon, that synaptic activity occurs at the synapse. The last component is information processing. How does the postsynaptic cell respond? And you really have two ways of responding. Either they're going to be depolarized and they're going to pass the signal on or react to the signal, or they're going to be inhibited. So that's information processing, the response of the postsynaptic cell. At the simplest level, individual neurons have many dendrites that receive neurotransmitter messages. Some of those messages are going to be excitatory, some are going to be inhibitory. So we have to consider the net effect, which is going to occur at the axon hillock, this region just before the first segment of the axon, into whether or not an action potential is produced. So in reality, let me say this just again, the dendrites, these little extensions, short extensions off the cell body of the neuron, they're going to be receiving a tremendous amount of stimulation. Some of those are going to be excitatory, depolarizing effects. Some are going to be inhibitory. So the net effect, if you take all the excitatory and all the inhibitory and add them together, you get the net effect. The net effect on the axon hillock determines whether or not an action potential is produced. Postsynaptic potentials are graded potentials that are developed in a postsynaptic cell. And again, they're in response to the neurotransmitters. And we have two types. Excitatory postsynaptic potential is EPSP. It leads to a graded depolarization of the postsynaptic cell. If you consider our scale, an excitatory postsynaptic potential is going to cause a depolarization effect, while an inhibitory is going to cause a hyperpolarization effect at the postsynaptic membrane. So a neuron that receives many IPSPs is inhibited from producing an action potential because it's been hyperpolarized. It's been, again, I ought to get a stamp for this since I draw it so often. If a cell is getting a lot of IPSPs, it's going to lead to an increasing hyperpolarization, and because the stimulation needed for that particular cell to reach threshold is increased, it's going to help inhibit action potentials. So in order to trigger an action potential, one EPSP, an excitatory postsynaptic potential, is not enough. EPSPs, and for the matter of argument, IPSPs, they combine through a process called summation. And in there you see the word sum, because all we're doing is adding up the individual effects. We can consider summation at two levels, temporal and spatial. And temporal summation, it caused by rapid, repeated stimuli at a single synapse. So the first stimulus here produces a graded depolarization, or in this case it could be a hyperpolarization, whether it is EPSP or IPSP. But a very rapid second stimulus is going to intensify. And if you get repeat stimuli, you could intensify that area, depolarize it enough to reach threshold. each individual EPSP or IPSP is it will only change your transmembrane potential by about 0.5 millivolts. And remember, in order to reach threshold, you're looking at 10 to 15 millivolts. So at the bare minimum, you're going to need 20 to 30 EPSPs in order to get that membrane to threshold. So through summation, we can get there. You just add up the individuals. So rapid fire EPSPs is going to eventually create enough depolarization to reach threshold, and at that point you get the generation of an action potential. So that is temporal summation, where you have, again, repeated stimuli at a single synapse. The alternative is spatial summation, and that's where you have two synapses that have overlapping fields. So here is your first synapse. Here is the field for your second. So you have this area of overlap right here. And through spatial summation, you get this stimuli, these stimuli that are going to arrive from multiple synapses, but because their fields overlap, you get you can add up the individual contributions of each individual. So you sum them together. So through spatial summation, you can also reach, action potential. You can also reach threshold, but it's going to require activity at all of those synapses with their overlapping fields. A neuron becomes facilitated as EPSPs accumulate, and that's going to raise the membrane potential closer and closer to threshold until a small stimulus can actually trigger an action potential. So again, consider your scale, -90, -70, 0, and 30. If you're sitting here at rest and you have rapid fire EPSPs, and that's going to continue to strengthen this depolarization, as soon as you reach threshold, boom, you get the generation of an action potential. It's propagated down the entire length of the axon, synaptic activity. Okay? So facilitation, a neuron becomes facilitated as the EPSPs accumulate. They're going to raise the transmembrane potential closer and closer to threshold until the final straw or stimulus can trigger an action potential. The summation of EPSPs and IPSPs. Neuromodulators and hormones can change the membrane's sensitivity to neurotransmitters, making them either more sensitive or less sensitive. And that's going to shift the balance between EPSPs and IPSPs. In summary, information is relayed in the form of action potentials. Neurotransmitters released at a synapse may have either an excitatory or inhibitory effect. Neuromodulators can alter the rate of neurotransmitter release or response of a postsynaptic neuron, and the response of the postsynaptic neuron can be altered by neuromodulators or other chemicals that cause facilitation or inhibition, activity underway at other synapses, or the modification of the rate of the release of neurotransmitters through facilitation or inhibition.